Okay, welcome back. Now, before we get into some detailed discussions around certain eukaryotic cell structures and their functions, I'm going to look at two principles here. The first one helps us really get to the heart of the issue of the difference in the structure and function of eukaryotes versus prokaryotes, and the second really lets us understand how we see different structures and functions in different cell types in the same animal. And we're going to focus on animal cells here as we do this. Okay, so this first point I want to bring up here is that the main difference between eukaryotic cells and prokaryotic cells is that eukaryotic cells are compartmentalized. This was a significant evolutionary leap, and the compartmentalization probably arose early on in the appearance of eukaryotic cells during some kind of endosymbiotic event where one primitive cell engulfed another primitive cell, and now we've got one cell inside another, and that's the first compartment, and that's probably how we got the evolution of the mitochondria and subsequently the chloroplast. But the point about compartmentalization is it lets you have specialization. It lets you have compartments at, for example, different pHs, where certain enzymes can be involved in certain types of reaction that you wouldn't want necessarily in the cytoplasm. It lets you keep certain events in certain places so that we can do gene expression in terms of transcription in the nucleus, along with all of the processing of those transcriptional products. So compartmentalization gives you specialization, and it was the major evolutionary jump that took us from prokaryotes to eukaryotes. So that's the first thing I wanted to bring up here. My second point, and we'll be done pretty quickly here, my second point is how do we explain the different structures and functions? Hey buddy, how do we explain the different structures and functions that we see in different cell types in the same organism? So think about what you have in your body. Your brain right now is processing you using neurons, and the inside of your mouth is lined with epithelials. Now let's go back in development to how we got those tissues. If we go back into development where we started off with sperm and egg, when they fused and we got a zygote, which is a fertilized egg, that cell has got a genome. Now, division through that idea from cell theory giving rise to all cells, one cell giving rise to other cells. That means that every single cell in your body, a few exceptions to this, every single cell in your body has exactly the same set of genes. So, why are these neurons on the left looking like neurons, doing the job of neurons of receiving information and creating action potentials and sending those electrical pulses down those long branches called the axons, versus the epithelials there, which are for protection and secretion of substances? Why do we have these two very, very different structures and therefore different functions when we've got the same set of genes? Okay, so the bottom line here is that all of the cells in your body have the same genome. Yes, there are some exceptions, but by and large, every single cell in your body has the same genome, but they have different sets of genes being expressed, producing different sets of proteins. So what we have is the same genome giving us different proteomes. So the proteome is the collection of all of the cells, all of the proteins in a given cell or collection of cells in a cell type at a particular time under a particular set of conditions. And that can change based on cells' responses to their environment, and it changes very early on in development when cells are becoming differentiated either as, for example, neurons or, for example, as epithelial cells. And so when you get different proteins, we get different structures that appear. Maybe we get an axon versus a flattened epithelial cell because of the different types of cytoskeleton proteins, which we'll talk about next time or the time after in the cells. So it's the proteome, it's the collection of proteins which gives you all of the structure and all of the function, which allows you to have this diversity of cell types that you need to build tissues and organs and organ systems and ultimately the organism that you are. So that's all I wanted to cover here. Two really important principles to take away from this. The idea that a major evolutionary jump was made when we had compartmentalization appearing, and also that you get different structures and different functions in different cell types because even though you've got the same genome in those cells, you have different genes being expressed, giving you hugely diverse proteins, and therefore a diverse proteome to give you diversity of structure and function. That's it. I'll see you next time.